a big occasion, and we are almost running the NMR workshop for the last 5 days, and we started with 2 of the legends, and now we are at the last working day, and then we are the experimental things coming. So we have another one, and legend of that NMR in India, Professor Chary. Professor Chary is currently director ICGEB-DBT. So that is now the big thing which he is known for. Professor Chary's name is listed in the history because he is the one who has solved the first solution structure of a protein from India. So that remains a credit. That's a big thing from here. So, and that is long back. So, and when NMR was actually developing, and that was a calcium binding protein, and it was a tough protein actually, because what you think, because crystal structure was solved and it was a trimer structure, and solution structure had its own domain structure, and it was 2 domains were tumbling independently, and that was thing which was not very much known that time, and he did those structures that finally all the reviewers had to accept this thing, and it is a landmark paper. Whenever the multi-domain protein structures are solved, this paper is always referred to that. He also did the work on metal binding, the calcium binding to the protein, how the sequential binding takes place to those proteins, and of course there are many other contributions to that, like automation and other things for which he is known. So he was that thing he did in India for. And another important thing about him is not only the NMR spectroscopist as running the spectrometer spectra. He himself is a kind of engineer, so he knows how in and out of the machine. So he, whenever there was used to be a problem, any hardware problem, used to rush to him for support and other things. So that's why I thought that his presence will be a good thing here to motivate all of you to learn how the NMR can provide new insights, new information, and how one can tweak the instrument or tweak the software, build a new thing. He has developed many softwares also for automated assignments. The initial one like Tata Pro and everything, which came from his lab. So with this work, I invite Professor Chary to start. Yeah.
Thank you, Neil. It's a pleasure to be here at ICGEB. I have to recall my first visit to this place. First and the last visit was in 1991. And precisely exactly 31 years afterwards, I'm here with you. As a part of this NMR group in TFR, we always had this habit of organizing at least one workshop every year. Okay, and we would really enjoy it, put our heart into it, and see that everybody goes at least with some message. And that continued. And last 2 years, it has been really little bad. This pandemic has taught us so many lessons, wonderful lessons, actually I should say that. Yes, we have lost some of our dears and nears, but it has done a great deal of good things actually. I don't think anybody could have taught as much as this organism has taught us. It has taught us how to behave, how to do anything and everything in a very different way. Okay, with that note, I should once again thank Neil for giving me this opportunity to be here. And he said that it's going to be a plenary talk. And now, to talk anything about NMR is not an easy task actually. Unfortunately, quite often we end up, any speaker, for the matter, even Neil or Mohan or anybody in the audience, if they have to talk, they always question where to start and how to start. And that is the sad part of it. And quite often, whenever there is a workshop, there are a variety of audience actually. Those who are just getting introduced, those who are already practicing it, those who are professionals. Okay. Now, I had this opportunity of taking a course advanced lab actually. He puts a question to the audience. I'm sorry, I'm taking some time. It's a very interesting thing to Professor Ernst starts his lecture first saying that how many of you want the course in German? And everybody raises the hand. And he knows, I mean, somebody sitting in the front row is not raising the hand. And then he puts another question, how many of you want really in German or in English? Then again, German or English, we should be very careful about that. And then he comes to how many of you want only in English? And then immediately he says that this course is going to be only in English. And this was very touching actually. And I'm going to dedicate this talk to him for the person whom we have lost, I mean, during the last year. Okay? And again, learning from him, now I gave the variety of audience here, those who are just entering into the field, those who have already started practicing it, and those who are professionals. But I will touch upon those who are at the entry level, and those who are stalwarts here has to excuse me if I am going too introductory. Okay? And another thing which I said, that when you cannot find real relevance to the topic of the workshop, okay? And he wanted me to end up with giving an overview, okay? And that is the spirit with which I have here, and I hope the next 40-50 minutes, I think you would enjoy what I'm trying to say. And immediately then, when it is an overview, I thought that I should coin it as esoteric, because I really enjoy, and I always say, I sleep, I eat, I drink, and I only do NMR and nothing else. And that's what I am. And if I am here, it's only because of that. Okay? So, my dear friends, those who are just entering, this NMR is an intricate. Sorry. I didn't. Yeah, it's fine. Yeah. So, what is esoteric? Okay, that is what you would enjoy in the next 40-50 minutes. NMR is an intricate and elaborate choreography of certain nuclei. They dance to the tune of the spectroscopist who is at the fingerboard, okay, or in front of the NMR instrument. They respond with mellifluous melodies which are not palatable to the human ears but carefully picked up by electronic ears to decipher their songs composed in the language of radio frequencies. So that is the interesting part of it. Okay? And I always start with this slide before introducing NMR. First and foremost thing, you should understand that it is in the structure and the dynamics. Understanding the structure and dynamics of a molecule is important to understanding both its biology and chemistry. And now I'll take you to the developments that have taken during the last 3 decades. I was to always show only 3 of them, but now recently I have added the 4th one also. So there have been 4 significant developments that took place in science. It's not that these are the only 4, there are plenty of them, and which I am going to highlight. I'm sorry for this inconvenience, it is coming on and off. Some problem? Somewhere? Am I doing anything? I'll better use this. Yeah. So the four significant developments are the sequencing of various genomes that spur the advent of genome projects. Another one is the NMR spectroscopy, and another one is cryo-electron microscopy, which have become a very powerful complementary tools to crystallography to unravel the structures of large molecules. And more recently, the DeepMind's AlphaFold has provided a solution to the 50-year-old grand challenge in biology. I want to highlight here this. I do not know, some of you might not have been born even at that time. Okay? In 2001 there were two seminal papers, publications, and which have brought us this post-genomic era. As of today, and a few days back I have taken the statistics, you wonder that there are 483,000 organisms which have been sequenced, and some of them are celebrating 22nd anniversary. In this post-genomic era, those who are born in the dawn of the 21st century, particularly 290 million Indians, I should say that, I'm sorry. There are some outsiders too in the audience, and I thought that it's going to be only India. This is with regard to the population of Indian population, which is around 1400 million today, okay, and 290 million have been born during this 21st century and started turning into 22 years. These millennials, of which some of you belong to crew with technology, smartphones in your pockets, and are emerging as aspiring generations with a lot of promise and potential, and are railing to take India into higher orbits. This another thing which is very interesting development that has happened very recently is this, what is known as DeepMind's AlphaFold protein structure predictions for almost entire human proteome, 98.5% of the human proteins, and 20 other key organisms are now available to accelerate scientific research in an unprecedented scale. It's a solution to 50-year-old grand challenge in biology. They never thought that they would see the day, but it is really remarkable. Another thing, I don't know, some of you may be looking at this data bank called Protein Data Bank. If you look at this structure that are being deposited, what you observe is that an exponential growth in it. As of today, you see that around 190,000 structures deposited in the data bank. And what is interesting for the young in the audience is that this exponential growth, if not for more than a decade or two, but at least in the coming decade, it should continue to grow. So what I'm trying to highlight is that if you want to take up this as a profession of unraveling structures, and maybe that one day you will definitely be depositing your own structure into this and contributing to this exponential growth. So the message that I want to convey here is that we are in an exponential world, literally in an exponential world. And most important thing which I want to highlight is that this is amazing actually. The kind of growth in the depositions coming from this electron microscopy is amazing actually. This is, I mean, I would say that even 5 years back, it was not believed actually. So, as you see here, I think very soon this may be taking over even crystallography. A faculty who joined our institute, who had got introduction to this cryo-helium, started saying that I don't need a diffractometer, and I was stunned actually. He wants to only practice cryo-helium, and this is the work of these people coming from, and the kind of resolutions that you have is this. I see here is almost a 1.15 angstrom resolution structure of a dilute water dissolved protein called apoferritin, actually 440 kilodalton protein, actually amazing 1.15 angstrom structure coming from electron microscopy. And this is another one which is 1.7 angstrom resolution, and this is another very interesting membrane protein. And this is stunning, actually. And on the other hand, I mean, this was shared by the faculty who joined us very recently, and he says that this kind of revolution is amazing, actually. From the start of preparing the sample to the getting the structure is just only 8 hours. Can't imagine, actually. Okay? So this is the world in which we are. So this cryo-EM is you can have structural information for complexes which are not crystallizable, okay? Particularly that's more important, as well as vital cellular context, actually. On the other hand, to address any infectious disease, I mean, this is the way to go for. I mean, I will end up my talk with that slide. And another important thing is that the title of this workshop, I mean, drug discovery, and in order to just highlight about that, even in drug discovery, this is going to be an important tool today. And these kind of structures, actually, the spike protein of the coronavirus with ACE2 could be solved in just 10 to 15 days. Can't imagine that. And with antibodies, again, in few days. So today you have the virus or its mutated form, and within a week you have the structure of not just spike protein with those mutations, but also even with the complexes with ACE2 or these antibodies. So my dear friends, what I want to highlight here is that we are in an exponential world. The number of biomolecular structures deposited each year in the PDB has been growing exponentially, and the number of structures unrelated to other proteins has not. And this is primarily due to the practice of the structural biologist. If I crack a structure of a calcium binding protein, tomorrow I'll just mutate it and then try to do, or complex it with something and then try and deposit it, and it continues. Not necessarily that every day, I mean, I come up with a new fold. Okay? And however, we also know that, and we should realize that the universe of the protein structure is definitely finite, and the chance occurrence of the structural similarity has become increasingly common. This trend appears to be definitely continuing to be positive for the task of deriving function from these structures, actually. So the message which I wanted to convey is that the rate of generation of the new structures ought to accelerate to even start keeping up the sequence information coming from these genome labs. When I said that 480,000 genomes have been sequenced, now imagine that on average, let us say that there are 10,000 genes or even 1,000 genes. You can imagine that, how much of sequence information that you have. And as a practicing NMR spectroscopist, I would always say that yes, NMR still has a role to play and will have a role to play, and don't be disheartened to see that exponential growth of these EM structures that are being deposited, actually. Again, remember that if anything is flexible, which cannot be crystallized, you cannot go to diffractometer. On the other hand, anything which is too flexible also cannot be imaged with this cryo-EM. Okay? So we should say that NMR will continue to be complementing results obtained by these crystallography. So now, having heard about 1D, 2D, 3D and dynamics, and that's what I heard from Priya while I was driving from INSA to this place. So I don't have to again reintroduce to you, but I will touch upon some very interesting trajectory of this NMR. Okay, the first and foremost thing, unfortunately, is not really recognized by a Nobel Prize or that sort, has come from a Belgian physicist called Jean Zenner. Actually, he's a theoretician, and in Ampere school, he seems to have presented to the audience saying that, hey, if you have a 90-degree pulse applied to this function, that function could be transformed into this function. So in NMR knowledge, I mean, this is a property of spin I, and this becomes the property of spin X. So what is needed is that this I and S have to be coupled. If there is a coupling, you have this kind of a term, which is really the property of spin I, and suddenly gets transformed into the property of spin S. And what is needed is a non-selective 90-degree pulse that excites both spin I and S. So this is the seminal work that has come from a theoretician. Unfortunately, it couldn't translate into an experiment. I'm sure, my dear friends, if at all you had to go out of this workshop, at least go with this message that if you have understood this, I think you have understood something about NMR. That's the beauty of it. So now, having done that, this particular idea was dormant almost for 3 years. Absolutely nobody was able to give a shape to that. Okay, and suddenly this gentleman who just returned from Viren labs after discovering the FT NMR technique in 1966, he joined Archaea back, and then in 1974 somehow suddenly silently shows to the world that yes, indeed, this could be experimentally shown by what is coined as a cozy experiment, a correlation spectroscopy where you can correlate coupled spins. And that's what was his ingenious experimental demonstration to the world. And that gave birth to what is known as 2D NMR. And just opposite to his lab, on a 5-story structure on a terrace, was the laboratory of Kututhrik, who started looking through the window and started seeing these colorful 2-dimensional experiments being plotted on a paper. There was no displays at all that time, and suddenly he said, hey, why should not we collaborate? And that's how they collaborated together, and suddenly he provided some sample and recorded such spectra and started deriving the structural information from that. So that collaboration went on almost for 15-20 years. Okay, so this NMR spectroscopy witnessed a sudden transformation into 2 dimensions in 1970s, and since then there have been several of them, and 2D NMR spectroscopy has provided a powerful arsenal of various tools for yielding information on various spectral parameters. And if I had to list out, it will be more than maybe 1,000 such pulse sequences, each one giving a unique spectra. Okay? And then all of a sudden, those who don't like your work always try to pull your legs. In a very like manner, people who didn't like all those fascinated 2D spectra or 1D spectra, they started saying that, hey, come on, what else? That nothing else, no more, what that? No more research. Okay. Okay, NMR stands for no more research. And everybody were disheartened, but these people never gave up, and that should be your attitude too. And that is primarily because when you go to, actually, if you crack today a 5 kilodalton peptide, you want 10 kilodalton, and tomorrow you want to crack another 20 kilodalton, and you want to just go on increasing your ability to crack larger and larger samples. And that is when he felt that, world felt that, cracking a 10, more than a 10 kilodalton protein is not an easy job. Yes, it is true. Why? It is because of overcrowding of peaks and decrease in the sensitivity, because as the size of the molecule increases, it tumbles very, very slowly. When it tumbles very, very slowly, what happens is the relaxation process will be so fast and your line widths will be broad and the intensity goes down. That means the sensitivity falls with increasing the sizes. And then people who wanted to laugh at started saying that these 2D versions will no longer take you anymore beyond 10 kilodalton proteins. So this is when again people questioned that what is the alternate. So there was a need for an alternate strategy, and that gave birth to what is known as a 3D NMR. So till then, the focus was mostly not that there was no recording of C13 or N15 spectra, but till then mostly it was proton NMR or looking at hydrogen nuclei. But suddenly they felt that we should also take advantage of the C13 and N15, which usually form the form of any given molecule, macromolecule. And that is when people started questioning that why we should do that. Primarily because those coupling constants, that single bond coupling constants, are very, very large. And that Zener's seminal idea is much more efficient if you have to transfer the magnetization from a proton to C13 or proton to N15, because these coupling constants are several fold actually, whereas proton-proton couplings are just 12 to 20 hertz or maybe 3 to 20 hertz. These coupling constants are 90 to 220 hertz. Okay, so the efficiency of this COSY, the coherence transfer from one spin to the other spin, becomes much more high when you have two heteronuclear spins. So that was one thing. Another thing is that the dispersion of the chemical shifts is pretty large. If you look at the C13 spectrum, it spans 220 ppm, whereas your proton spectrum is maybe 0 to 20 ppm. Okay, and when N15 you will have almost sometimes it is 40 ppm. Okay, but however, again, nature has put very interesting condition there. Hey, you will find only 1 out of 100 is C13 and only 1 out of 300 is N15. So what you have to do? And this is where when you need to have an alternate strategy of making these biomolecules, proteins or nucleic acids 100% C13 and N15. And this is where the biologists came into the game. What did they show? They showed that you can take a plasmid DNA, a small DNA, and plant your signature DNA in that and close it and then transform into the bacteria, which grows overnight or in a few hours. And the fact that this plasmid DNA multiplies several folds as compared to the chromosomal DNA, actually it will have several multiplications, and thereby the protein that is coming out of that will be several fold, and that is why it is called overexpression. So the protein gets overexpressed, and when that protein overexpressed, you have to monitor these bacteria when they are growing. What they have done? They have done in such a way that this bacteria bacteria eats only N15 labeled ammonium chloride or C13 labeled glucose and enjoys, and there is no other nitrogen source or carbon source, and the fact that it eats only C13 unconsciously, everything that it produces in the form of protein will be definitely a C13 100% labeled or N15 100% labeled. The choice is here that you can cleverly only label N15 or only label C13, or you can simultaneously label both of them, or you can feed the bacteria 50% labeled and 50% unlabeled. So you can do uniformly, you can do fractionally, you can do both of them together and all that. So what it means that you end up transforming this polypeptide chain, which is made up of these nitrogens and carbons and hydrogens, all of them into NMR active nuclei called N15, C13 and proton. And the moment you have that, now you go back to Zener's idea and use that idea to transform or transfer the magnetization from one spin to another spin, and that give birth to what are known as these double resonance and triple resonance spectra. This is what is called, there was a parallel renaissance in biological chemistry and NMR spectroscopy, which witnessed a collaboration between the two to unravel the scopes of one from the merits of the other. That happened in 1990s. So, if you have a polypeptide chain and if you have cleverly fed the bacteria with only N15 labeled ammonium chloride, you have this kind of a combination. So, what you have is now you can transfer the magnetization of this spin to this spin by using that Zener method, and that gave birth to what is known as N15 proton correlated spectrum, that is called homonuclear single quantum coherence experiment, which can be recorded in less than 5 minutes if you have a sample of 1 millimolar sample. So this gave birth to a new cozy experiment called heteronuclear cozy, and you have this kind of spectrum where one axis is N15, other axis is proton. As you notice here, this is just only amide proton region, which is just 4 ppm, whereas here you have almost 30 ppm. So there is a large dispersion. Okay, very interesting one. So because of that, now all the overlap problems have disappeared now. As you notice that definitely this will be having 200 odd dots. That means you can easily assume that the protein must have been a 200 amino acid residue long. So this further has led to what is known as development of 2D, 3D and 4D, and here in this what you do, you can transfer the magnetization from proton to N15 and again to proton, and you can do some kind of a nosy experiment, or you can transfer from proton to N15 to C13 of the sequential neighbor or the cell itself, and you can go on recording this very interesting double resonance spectra or a triple resonance spectra. And now you have several ways of transferring this magnetization, only to the C alpha and C beta, or only sequential C alpha C beta, or only to the carbonyl carbon, and you have a variety of such experiments that you can record, and then you can get information about the chemical shifts of protons, N15s and C13s. So typical three-dimensional spectrum looks something like this, where you have one axis as proton, another one is N15 and another one is a C13. And one plane taken out of this will look something like this. And you see that this is very sparsely filled actually. So there is no overlap at all. So in principle, it becomes much easier for you to analyze the spectra. And that was not the end of the thing. Simultaneously, there were the technological revolutions, and this is a pretty old slide, though again the sensitivity of the NMR spectrometer started going on increasing year after year, and primarily it is because of higher fields. So people were not satisfied 600, 700, 800, 900 and even 1 gigahertz NMR spectrometers, and added to that one high temperature superconducting probes where the NMR coil is kept at almost at liquid helium temperature, and whereas the sample that you keep was at room temperature or desired temperature. And that was the very interesting development that has happened, and this low temperature coils and preamplifiers and better RF technology led to this. And just not the isotopic labeling method nor this development, but it is the relentless efforts of certain groups continuing to work and break the limits and continuing to push this molecular weight to higher and higher, and went on. But again, people who wanted to show fingers had some fun in showing this kind of a graph and saying that, hey, all your NMR structures that are being deposited are less than 25 kilodalton, and come on, you cannot keep your head high and collar high, and that's it. Now, why? Again, it is inherent with the molecules that you play, and unfortunately they tumble away so slowly, the relaxation process are so fast, lines become broad because the signals decay. And again, people started questioning how to overcome that. So, why such low occurrence of these structures? Primarily, it is because of the inherent nature of the macromolecules. So, what do they have? As the size increases, there is what is known as chemical shift anisotropy comes into picture. There is what is known as dipolar and dipole-dipole interactions, and these are some kind of a relaxation processes which kill the signal. Okay? So, the NMR signal, instead of decaying so slowly, it decays fast, and thereby the phototransmission of that is a broad signal. So, now people were clever enough to identify and see an opportunity to see that the fluctuations due to dipolar fields and fluctuations due to CSA either add up or cancel. When they add up, naturally it will be much faster relaxation, so naturally the signal will be broad. On the other hand, if they can cancel each other, as I have highlighted here, what happens? Their effect, the net effect will be less, and thereby the signal will be sharp. So, as a matter of fact, when I was showing the N15 proton spectra, though there was only one dot, actually it is a combination of 4 dots actually. So, out of those 4 dots, one dot, one component is sharp, whereas other 3 are broad actually. So, people were clever enough to pick up this sharp one and discard that, and that is how they said that, hey, we can break this problem, overcome this problem by what is known as transverse relaxation optimized spectroscopic technique, where that sharper signal, sharper component is selected and the other components are suppressed. And that is how this kind of picture, where people would have laughed at and said that, hey, you cannot crack this spectrum or the structure, started had a law, lost law, thereby, you could see this kind of signature and very nicely. And this led to even cracking the higher molecular weight proteins, and some people even proudly said that even 100,000 kilodalton protein also, a oligomer, could also be cracked There's another thing, is there any other alternate? Is it that all? Not necessarily. People started saying that why we should be after protons, why not just be something else? Let us correlate only N15 and proton, or N15 and carbon, or carbon-carbon. Indeed, it is possible. Why? Because the relaxation enhancement by paramagnetic methods is like that. I mean, this 1 over T2 dipolar is directly proportional to gamma I square, which is the observable, the nuclei that you are observing it. Okay? The fact that this gamma S is 1/4 of that, in principle, by detecting the carbon, you can enhance this relaxation processes. I mean, reduce the relaxation process and then get the sensitivity by a factor of 16 actually. This is amazing actually, and people were able to record even this kind of spectra of a ferritin which is 480 kilodalton and still get a lot of spectral information that is desired. This is C13 correlation experiment. So the journey went on and on. My dear friends, it is not that you just take the sample and record one spectra and get the structure out of it or derive the structural information. No, certainly not. You have to record a suite of spectra that gives different information, and for that, naturally, you have to analyze all the spectra that you collect, and analyzing that became a cumbersome, tedious, and that time people thought that why we should not try to automate, and this is where another small diversion took place, and those who are proficient with software development could do this. So manual assignment is tedious and time consuming, hence there was an automated approach was needed, but for that you have to have very good input of peaks, and also there should be reliable classification of individual spin systems. So for that, what you need to do is that you have to have some kind of artificial intelligence. You have to analyze the so-called big data that is coming out of these different data banks. There is a data bank called Biomarker Bank, BMRB Bank, in which rather everybody deposits the chemical shifts. So simple analysis of that chemical shift data can tell you that, hey, all the alanine resonances of C13 and C13 beta carbon and C13 alpha carbon will be somewhere here. Oh, given any such triple resonance spectra, whenever you see a dot in this region, and you can attribute that it definitely has to come from alanine. So this is what I call it as artificial intelligence, and you can train your machine that, hey, if you find this data at this point, this should be attributed to be coming from this particular amino acid residue, and if it is coming from there, something else. Based on that, I mean, you could teach this machine that you can classify these different amino acid signatures into different regions, and you can in principle do automatically. So you don't need to sit and analyze the data. My dear friends, though I might have written in this paper that it could be done in one day, it is not always easy and straightforward, but it is definitely better than what it could be if this software is not there in your hands. Okay? Then suddenly again people started saying, hey, my dear friend, I don't have a spectrometer, I don't have so much time to collect so much data, and again there was a bottleneck. Now our cricket started turning into T20 matches actually. Nobody wanted to have 5-day play or 1-day play, they wanted to finish in 4 hours, and that was the time when people started saying that, my dear friends, I cannot sit and collect 10 spectra and sit and analyze and get the structural information. I want to do somehow a something which I can collect in just even 20 minutes. Is it possible? Yes. And that has been shown by this Archaea and Transverse by saying what is known as simultaneous acquisition, simultaneous sampling of these along 2 dimensions of a 3-dimensional spectrum, and got that 3-dimensional information in 2-dimensional experiment, 2-dimensional space, and showed to the world that it is indeed, it is possible to reduce the time of the NMR spectral data collection. Okay. So, in principle, a 3D information can be translated into 2D, and 4D information could be translated into 2D and 3D, and there were several other alternates which I don't want to highlight now because of the paucity of time. So, I try to give an overview, but certainly not, this is not just everything actually. For every issue, every problem, you will definitely find a definite solution. Okay. Only thing is you have to have the tenacity to see what is needed and what is not. So other alternate approaches are, I mean, you can just not only just use proton and C13 and 15, sometimes you may have to deuterate the sample. By deuterating the sample, what you do, you enhance the sensitivity of the spectrum. Why? The relaxation process are slowed down actually. If you can partially deuterate, you get a more sensitive spectra. On the other hand, you can do uniform or selective isotope labeling or fractal isotope labeling, or you can only do this deuterium labeling, or there is what is known as stereo array isotope labeling, where suppose if you take an arginine, you have CH2, CH2, CH2. There is a abundance of protons actually. What you can do is that you can replace one hydrogen with deuterium, and in the next CH2 another proton with a CH2, then what happens? The relaxation process of the attached carbons will be very, very slow. Thereby you enhance the sensitivity of that. So there are several things. I think those who are keen can spend some time in the evening or the coming days in trying to look at that. So the message is that the rate of generation of the new structures ought to accelerate to even start keeping up the sequence information being generated now, which I have said already. Okay. So it is indeed possible, whether it is by X-rays or NMR or electron microscopy. Now, coming to this drug design data, which I thought I will just touch upon with some few slides. I am not showing you any spectra. Okay. These NMR screening methods constitute a very reliable and valuable tool for identification of small molecules and hit to lead optimization. A library of compounds is screened for finding the hits and that can bind to a specific target, followed by their validation, for which NMR binding assays can be of high efficacy. So what are the methods? So there are two kinds of methods actually. These NMR screening methods have broad application during the lead optimization process. This is primarily because hits obtained are generally weak binders. This is the advantage of NMR spectroscopy actually. It is easy to bind, to see even when the binding between the ligand and target is very, very weak. Okay? NMR methods can provide high quality structural information for complexes of weakly bound ligands, and NMR methods for lead optimization also have two modes of detection. One is called target detection or ligand detection actually, or that means focusing on target ligands or ligand residences. So there are two things actually. The target resonance based screening means, I mean, there are two popular techniques. I will just touch upon only two methods, which is basically, what is known as chemical shift perturbation. What is chemical shift perturbation? In chemical shift perturbation approach, the information on the intermolecular interaction between the compound, the ligand, and the protein, which is called target, can be extracted based on the chemical difference between the free and bound protein. So what you have to do, you have to record once the spectra free one and one in the bound form actually, and the difference immediately tells about that, and that is what exactly is called chemical shift perturbation. So for this method, the target protein needs to be labeled. That is the very interesting part of it. You can label only the target protein and then add the ligand and then record the spectra and then see what is the effect of it. And I would like to highlight one of these, just one spectra that I wanted to show you. This is a protein which is called helium. Okay, it is a crystalline domain protein actually. Two Greek motifs. In the absence of the calcium, actually it is structureless and it looks something like that. And you just simply add calcium and suddenly you see this kind of spectrum. You see these two lines here, the two methyl resonances prop up here, and the dispersion in the amide region is amazing here. And look at this apo helium. So my dear friends, whenever you come up with any new protein in hand and go to NMR lab, you will be disappointed first thing and foremost thing, and if you see this kind of a HSQC spectrum and you see that I have spent 2 years and I have got nothing out of it, and then you start even crying actually. I have seen several students doing that in my laboratory, and now a fellow friend comes and says that, hey, why don't you put a little bit calcium and then see what happens, and suddenly he adds the calcium and then suddenly the spectrum transforms into this, what I have shown on the right. So immediately everything is gone and he's taken to a movie and a party and whatnot. Everything happens suddenly because they are sure that they are definitely going to get a publication out of it. And this is the life of an NMR spectroscopist, and you have to live with it. And the pressures will be high even to the guides too, not just only the students. Okay? So this chemical shift perturbation method has many advantages. Apart from being very reliable and reproducible, it can provide crucial structural information on ligand binding site. It can detect compounds with low to high affinities for target. Am I taking too much time? Okay. It can detect compounds with low to high affinities for the target, and the dissociation constants also can be measured, and if the structure of the target has been elucidated using NMR, the ligand protein distance can be derived from the NOE type measurements like what I have demonstrated. HSQC, NOSI, HSQC, toxilec kind of experiments can be done with, and then you can get even the structural equation and the kind of interaction that is there between the ligand and the target. Okay, there is another thing actually. Sometimes when you go from one molecule to the 2 molecules or 3 molecules, actually the size goes on increasing and the spectral complexity becomes increasingly large. So at that time, what you do? So there have been proposals that to concentrate only on methyl region, not necessarily the entire spectrum. Okay. So there is an advantage. I don't want to read that, and those who are keen can try to look into that. So, but there is a way to go about by just only concentrating on methyl regions. But to see these methyl regions, what you have to do, I mean, there are some special methods to label these only methyl groups where the secretin can be labeled. Okay, there are some precursors for getting that kind of protein, and you suppress all the other part of the spectra and you only highlight those things and get that information. On the other hand, the second screening method is what is known as ligand-based screening method. Here the ligand is supposed to be the smaller one, so naturally, so it is much easier to do it, and you don't need to label the large protein molecule ligand. Okay, and target, and then you record the spectra and only focus on your target resonances. So in the bound form, in the unbound form, and usually in recording the spectra actually will take very less time, because you will be recording either 1D or 2D, and there is no upper limit for this. Okay. There are other saturation transfer difference experiment, and there is also what is known as a diffusion edited NMR experiments, where you only look at the diffusion characteristics in the bound form and the free form, and you can get that information about these interactions and interaction surfaces. And there is another thing, what is known as transferred NOE, where you can try to look at the what you can saturate and then try to see the NOE, and the process rather from protein you can transfer the NOE to the ligand, and then you can get that information too. So if the ligand has low affinity binding or weak interactions with the target protein, transferred NOE spectroscopy method can provide an excellent alternative. Okay, so this is what I wanted to highlight, some of these identification of the interaction between a ligand and the target actually. So I will just conclude by just flashing two of the structures which have come out of my laboratory. Okay, and we were working on what are known as crystallines. I will just tell you how you can just go from sequence to structure and enjoy that. Okay, so here I wanted to just show you this, looking through the structure. This islands is an example of nature's fascinating conception. Okay, and this transparent mammalian islands is made up of proteins called crystallines, which are classified into two distinct superfamilies called alpha crystallines and beta gamma crystallines. Beta gamma crystallines are two different kinds of crystallines together put under one superfamily, primarily because their structures are similar to that and sizes are also almost same together. So there are different classes of these crystallines, and they are stable, long-lived from birth to death. I mean, you still have the same cell in your eye and recruited for dual functions, and evolution significance is that there is a gene duplication and gene sharing and fusion. They are typically made up of what are known as Greek motif, which is nothing but antiparallel beta sheet with 4 strands. They fuse together to form a two Greek motif domain, and then further duplicate and fuse and form such beautiful 2 domain 4 Greek motif structure actually. And this is what our islands is. Okay, and the members of this family have been identified in 2 or 3 domains of life, namely bacteria and eukaryote, but they were not when we took up this project in Archaea. So we thought that the Archaea being the oldest organism on the planet Earth, we thought that why should not we see whether we have the signatures of this eye in that, and then we tried to see and question ourselves. And to cut short the story, when these archaeal organisms have been sequenced, we tried to search for the signatures of the crystalline protein, and suddenly found one sequence of this kind in Methanosarcina, which have these crystalline signatures, and tried to overexpress that protein and looked at its sequence homology with other known islands proteins. Here, I mean, we could find to the 47% to 62% of that, and we did NMR work on that, and we tried to see calcium binding properties, and then eventually got such beautiful structures which indeed were similar to the islands protein structure actually. So that is when we declared to the world that, I mean, our island signatures are even present in the oldest organism called Archaea, and this is the one which is in the absence of the calcium and which is little flappy, and in the presence of the calcium it takes an ordered conformation. And to understand any kind of interaction between a ligand and a protein, protein. I mean, this is the wonderful experiment actually. What you do, you just add calcium ion, which you can call it as a ligand itself, okay, and see what kind of a transformation it takes, and where it goes and sits, and how it sits, and what are the binding, what are the coordinating residues and all that. And once you are able to make the calcium binding, you can even do with some kind of a paramagnetic titration with paramagnetic ions, and then see which broaden and which don't broaden, and from broadening them you can conclude that, oh, this must have been coordinating with this particular residue and all that. So then you can map the calcium binding sites, and then you can use other molecular phylogeny to show that it indeed these archaeal proteins are ancestors of these islands proteins and all that. And when you overlay with this C-terminal domain of the gamma crystalline, and it was, I mean, the RMS was 2.9, and that clearly shows that it indeed is has the same structural information as that of islands, okay, and all that I have, in summary, I have already shown to you. And another one very recently, just 2 slides to conclude, and we did show to the world, I mean, what kind of a single point mutation which leads to severe infantile cataracts actually. It is not age related actually, it is genetic actually. It has been observed in 3 generations in the China family, okay, and shows that that single point mutation of 57 from G to W, and this is the pathetic situation actually, okay. As the child grows, he becomes blind, and we try to again go from the sequence to structure and show to the world that indeed unusually this protein, I mean, the question that we addressed was that what are the structural changes induced by this single point mutation, and we tried to hypothesize that the structural changes due to this mutation may be propagating through the 3D structure and providing insights into the mechanism of cataract. So human gamma S crystalline is again not crystallizable, and that's the reason we took up. Otherwise, I would not encourage any one of you to come to the NMR lab if you can crystallize it, because it is much faster to crack that. And we did several NMR structures and then showed to the world that this unusually tryptophan, which is hydrophobic, is exposed, and people cannot believe that. Indeed it is, okay, and that has indeed ended up in concluding that is leading to the aggregation and thereby forming the so-called cataract formation. So revisiting our initial hypothesis, the reframed conclusion was that the structural changes due to the mutation propagates through the only N-terminal domain without disturbing the C-terminal domain and leads to this cataract. Now, at the end, I want to pose one simple question. If you want to design a drug, is there an alternate, or NMR is the only alternate? Okay? And surprisingly, this is what I have learned very recently, that when Omicron, the first known confirmed Omicron infection was on 9th November 2021, and Omicron variant was first reported to this World Health Organization from South Africa on 24th. WHO designated this Omicron a variant of concern on 26th, and within 9 plus 4, 11, 13 days, okay, plasmid containing the Omicron spike protein gene got synthesized, and then on 16th of December it was published in Science, okay. Can you imagine now? Now, we have this wonderful workshop. You may have sleepless nights, but you want to go to EM, cryo-EM lab or NMR lab is a big question mark actually. And then I was trying to learn, I'm sorry, don't ask me too deeply about questions about these frames. This spike protein, which is complex with this ACE2, has this beautiful structure actually. This is a little blurred and this is even blurred, and one can now focus only on this portion of that and improve the resolution to this extent. And having got to know that this is the kind of interaction that is happening between the spike protein and this, now focus on this, and the kind of resolution that you have is this. This is amazing, and our journey is exactly 7 days. Okay? So, Omicron's molecular structure could help explain its global takeover, and they could answer everything, its functional characteristics. That's the beauty of it. So, my dear friends, at the end, I always try to show this, which I have borrowed from Professor Ernst, where he showed these growing gardens of physics, chemistry, biology, and medicine overlap, and at that intersection is NMR, which stands tall, and still, in spite of this cryo-EM development, still keeps its head high and has, it will continue to help you to understand and give a lot of information. I'm sorry, though in my abstract I did say that I will talk about dynamics. I couldn't talk because of the cost of time. When I was putting all this story together, I felt that it is too overwhelming, and at the end I have to say that unraveling the conformation of biologically or chemically important molecule not amenable to crystallography or even cryo-EM, I should say, I should have written that, and studying their dynamics and interaction with other molecules to understand structure function relationship is a challenge, and it is here where the NMR is an indispensable tool, and it's fun doing either 1D experiment or 2020 experiment, and always focus on any material research with a symbiotic relationship between the theory and experimental proofs. And this I could not have been in front of you without the contributions coming from students and the postdocs and my collaborators. And finally, I should say that those who are keen to know more about the subject, though this is written in 2008, it's still valid and it has very nice information for people who are just entering to the field. I would strongly recommend. It's 14 chapters, 500 odd pages, and little expensive though, but even PDF versions are available and you can choose that. And this is what Neil was telling about the calcium ion protein, which was solved for the first time in the laboratory in TFR. And thank you for your kind attention. I'm sorry if I've taken a little more time.
Thank you, Chary. It's wonderful to hear our story. It's a wonderful story. It's a journey of how it started and how it is developing and how it will develop probably in future. Of course, NMR means you talked about detail, and it's a bit challenging currently because of the current cryo, but things will evolve nevertheless. Because what I think I saw the last slide from Professor Ernst, NMR is like how the goddess Durga was there with arms from all physics, chemistry, biology, and medicine. So probably that way things will evolve. But of course, leave the question of the cryomes. With Dinakar here is one of the earliest guys who has worked with EM systems on viruses. So probably he may have some comments to share on that, how the things might be in future. Cryo versus NMR.
Something that you didn't speak, dynamics. you can't project that by X-ray diffraction and cryo-EM. Cryo-EM and X-ray diffraction are not different at all, except that you can do crystallization in computer in case of cryo-EM. Otherwise, it's all same. But dynamics we can see in real time.
Yeah.
Sorry. Questions, yeah. Pawan.
Sometimes, when you are making recombinant proteins, you get some aggregates. And you have to add either non-any detergent, or you have to add little urea or some other detergents. Now, sometimes it's very hard to really remove those aggregates. You have to add these reagents. Is it NMR can help to solve these kind of structures also?
Yeah, there are issues. It's not only that. Even salt can play havoc. Okay, and some proteins are soluble only at high salt concentration, for example. You have to live with that. But there are some probes which are designed to take care of this high salt concentration. Okay.
I have another thought. So in biological fluids, so if you want to really apply and to study the difference in the disease condition versus non-disease condition, is NMR becoming an instrument that can be useful?
Degenerating. What is that?
Let's see effect on drugs. What is happened to the
Effect of?
Drugs.
Uh-huh.
Mike.
Or what is happening in the bloods.
Uh-huh.
So some of these things, if you want to find out as an NMR as a tool, are there ways?
Yeah, I have not shown some spectra there. In principle, yes, and that's the hallmark of this whole workshop. That's what has been, I think, discussed at length about it. Yes, it is indeed. In the presence of what you have to do is that you have to record the spectra in unbound form, in the bound form, and then definitely you will see there are certain significant chemical shift differences, and those significant chemical shift differences will be only part of the molecule. Okay, so from that you define where it is going in binding. Is that did I answer your question?
I was trying to think of that as a diagnostics means like spectroscopy. No, it's very difficult to sometimes really deal with this kind of
Mm-hmm.
There are alteration, there are changes, or are the drugs acting. So that NMR can be made into the diagnostics kind of. Can we do that? Or
Anything which is observable is it is. Okay, I think inherently if there is some solubility issue, then there is a problem. As long as it is soluble and you can create a homogeneous complex, and you can definitely record the spectra, and you can get the interaction regions very clearly without any problem. There are plenty of methods for that.
Yes, great talk actually. I was talking to Neil actually. Very nice overview of the NMR, that's what we are talking. So coming to on just clarification that that crystalline, that protein that you said, where the mutation the glycine to tryptophan, where tryptophan is largely exposed, and that is leading to the probably aggregation, right? So what is actually like a experiment that when you do you probably see the aggregation, what is the concentration of the protein in the eye actually, the way that they are comparable, and that can be the conclusion we can extend it to that sort of.
In eye it will be much more than
Much more actually.
Yeah, much more than that. Yeah, it will be much more.
Mm-hmm.
Yeah.
So that is leading to a
Aggregation and then
mm-hmm.
yes, precipitation. Yeah. 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 That's how. That's what I was just go from sequence to structure and then get to know about it.
Thank you, Professor Chary. Very nice talk. Just, I was wondering whether, what could be the function of M crystalline, which you said is conserved in Archaea? Because
I mean, so far it has been identified as only structural protein actually, not been given any. I mean, there are still studies going on.
Yeah, because this folding exactly like
it's called only structural protein.
Yeah. So function is not.
A function is not known. Whereas alpha crystallines are called chevrons, as when called heat shock proteins.
Yeah, nice talk. So I have a different question in a sense that you have talked about quite a lot about proteins. What about the nucleic acids? Let's say, like the way that you were showing, like alanine has this kind of chemical shapes. In a sense, for RNA especially, is it possible to really characterize that way, that if you have that kind of chemical shapes with this, maybe we can try to predict some of these RNA structures? Is there any kind of a...
Just chemical shifts? Yes, sometimes it is possible.
The whole structure, if we want to predict?
I mean, see, to get the structural information, it's not just only chemical shifts.
Sure.
There are NOEs, there are hydrogen bonds, there are several other constraints that people collect a lot of information and then end up doing this structure. Yeah. No, I didn't get you. What exactly you were saying?
So the thing that you were saying, that with this 2D plot, where you have the specific regions of that, I think C13 alpha and beta, and you can actually predict that, okay, if you have that position, then you know that that's the alanine that is there in your structure. So in a sense, a similar kind of similarities can be even drawn in nucleic acids, let's say.
Yeah, that information that you are deriving is only chemical shift information from that spectrum. Okay, that chemical shift information even has local information. So you can say what is the torsional angle, whether it is part of an alpha helix or beta sheet, that you can predict based on chemical shift.
Okay, but that can be applied to also the NMR, the RNA structures.
Yeah, exactly. Yeah, see, basically the number of constraints that you use per residue, if there are 20 or more, you get a very high resolution structure. So aim is always to get 20 per residue. So that chemical shift value may be only 1, but there are other 19 other parameters which are also contributing to the definition of that particular structure. Yeah.
Thank you.
Hello, Professor. Yeah, thank you for the nice talk. So my question is, you described the AlphaFold as one of the main achievement in last 3 decades. So what's your view how it is going to affect, or you can say affect the field of structural biology in coming years?
What will be the effect of AlphaFold on the structural biology, basically?
Okay. Now the question is, you see, any theoretical work, I mean, you would not get a Nobel Prize unless and otherwise it is experimentally proved. Okay. If there is no information, there is no information. If there is some information, I mean, I would say that there is some information. Okay. So if you are tracking something, at least you can just see whether it is going hand in hand with this prediction or not. If it is going, then you jump and then say that. But you would also give a fillip to him then, okay? He will share your prize. So till then, it will be only, some kind of a joy, that's all, nothing else. Not beyond, I don't know whether they will be blessed with a Nobel Prize or not. Can you answer that question? Do you think that? I doubt. Any theoretical, it's something like string theory, no? string theory, people are waiting for somebody to demonstrate. Corpus like people, I mean, no, but the thing is that is happening because, I mean, experimentalists have proved that they are right.
That's the.
Yeah. This is what I am saying. If suppose say today I am publishing a calcium binding protein, okay? In parallel I can do whether that particular sequence can give some structural information and then correlate it. So it is something like justification of your own structure.
And others who were trying to develop three-dimensional structures simply computationally and wanted to make crystallography redundant, they have actually not succeeded by themselves, but they made crystallography faster and richer. The crystallographic refinement at the rate it happens now, or even now cryo-EM refinement, would not have been possible without incorporating those energy parameters that they developed.
Yeah.
So these fields are not competing with each other.
No.
They are actually
Complementing.
feeding each other.
Complementing. As a matter of fact, G.N. Ramachandran is one of the best example actually. He showed to the world about the Ramachandran map, but unfortunately, he couldn't be recognized. This is a very unfortunate thing to happen. Yeah, I mean,
Thank you, sir.
this is almost similar to that.
Again, one question, whether NMR or cryo-EM, which will give more number of novel folds. You showed the chart in the beginning that so many PDB structures have been deposited and it is rising exponentially. Whereas the fact is that not novel folds are being added to the PDB database. So do you think that there will be a race between NMR and cryo-EM for discovery of novel folds?
See, there are two kinds of, I mean, how I look at it. One is that for development of a technique, okay, I mean, already it is rewarded. Now, if suppose he uses the technique and answers a very challenging question, and then there is a possibility. Till then, I don't think, even, I can't say it today, but maybe some new fold, or which has a significant influence or answer some important challenging question, then there may be.
I mean, just few days back only, they synthesized a novel fold for the plastic degrading enzyme. It's completely artificial. The protein doesn't exist. It's a completely artificial. Taking the cue from the prediction, experiment, and all these things, they synthesize completely a new enzyme. No, yeah. They are from that to degrade the plastic fat. This is the most efficient enzyme. I think 2 days back. So with that, I thank and I request our director, Pradinakar Salunke, to give this one.
Oh, it's nice. Thank you.
This is a Indian calendar.
Oh, thanks.
Yeah, this is a real Indian calendar.
Thank you.
It's the first month.
Thank you, thank you. Yeah.
So all of you are invited for dinner at 7 o'clock, and the certificates will be distributed by our director.